Are there any barriers? Are there any challenges to closer collaboration that need to be overcome? And if so, how do we overcome them?
On the clinical side, I think the key word is time. We are pressed with time. We have our patient to take care. And sometimes we do not spend time to collect the blood, to explain the patient why we desperately need an extra biopsy. Sometimes we run into problems, ethical problems, in asking the patient if he is willing to donate a biopsy. Sometimes I have personal problem in asking a patient, because I ask myself, would you do an extra biopsy? You really have to have an internal, incredible drive toward the science to donate such a personal devotion to this. So that's one barrier. What would be the barrier from the laboratory?
I don't like to think of barriers. I think I like, when I have a problem, have a solution. I think that this organization, ESMO, and its collaborating organization in EACR, they can play leading roles in dealing with things that for example, the European Clinical Trials Directive and its practical implementation, the data protection, draconian measures that were going to be passed in the European Parliament. And I think our societies, ESMO, EACR, working together, can really play an influential role in overcoming, if you want, those regulatory barriers. What you were talking is more cultural barriers that might occur, and I think that requires people, it requires leadership, it requires commitment, and ultimately it requires a focus on patients. If you focus on the patients, as you were saying, the teams will work together.
Give us an example, if you would, of how a collaborative approach has helped in your work.
I trained at Yale in terms of preclinical experience, and I was working on these crazy cell lines that didn't really mean too much to me. And I was studying the mechanism of action of fluorouracil, one of the most widely used agents in colorectal cancer. And I came after a research that essentially provided evidence that if you give it for short term, this drug works in via RNA, and if you give it for prolonged infusion, it works via DNA. Now, through collaboration with the clinic, the Degramon regimen was developed based on this. So now we do FOLFIRI and FOLFOX based on those crazy little cells. That's a good example, I think, because almost every patient is being treated with FOLFOX, FOLFIRI, and biologics.
The way we have developed drugs until very recently, and you just gave an example of that, was using cell lines. A lot of these cell lines have been in the lab for 30 or 40 years. They have adjusted to plastic. They're very distant from what patients are. So increasingly, we're using patient explants as an alternative to cell lines and as a preclinical development platform. And, you cannot do that unless you have collaboration with surgeons, pathologists, nurses, consenting the patients, getting the sample live to the lab, implanting it in a mouse or putting it in culture in a petri dish, and then using it as a platform to screen for compounds and screen for new drugs. You can't do that without collaboration.